Angela Koehler, and I have the privilege and pleasure of chairing the future of health and life sciences. I do not have the pleasure of following Sally Kornbluth because it's always a challenge to follow Sally. But luckily, you gave the vision statement for HEALS, so yeah, that's great. Yeah, so I will have the pleasure of introducing three speakers who are thinking about the interface of health and life sciences and nanotechnology. And so I think there will be a slide that will show their faces, but we have Anna Jaklenec, who is at the Koch Institute. We have Jagesh Shah, who is from Mirai Bio, and Gio Traverso just snuck in, kind of. He's a professor in mechanical engineering. Let's see. So Sally mentioned the vision for HEALS, and what we're really doing as one of the presidential strategic initiatives is trying to think how we can build a community with folks across campus, including all 5 schools and the College of Computing, to try to tackle some of the most transformative problems in human health. And that includes artists, that includes people who are thinking about ethics, that includes our colleagues at Sloan, but also engineers and scientists. There's some guiding principles associated with how the different strategic initiatives that you're going to hear about today, you're going to hear about manufacturing, you're going to again hear about life sciences, and you're going to hear about climate. There's some guiding principles for us, and that is, again, trying to bring in community, trying to undertake open source strategies. There's Sally, I won't repeat. Trying to be people-focused, as was mentioned, trying to develop trainees across MIT. We really want to build communities. And I think also important for this audience, all the strategic initiatives are thinking about how do we engage our colleagues in industry at the earliest stages for partnership and collaboration. And then we have an institutional commitment as these strategic initiatives to work with you to try to identify funding sources to actually enable that research. So on the HEALS side, here's sort of our pillars within HEALS. We're focused on developing talent. So for us, that means trying to develop funding sources for trainees across the board. We care deeply about entrepreneurship and trying to take our innovations from within the Institute to the wider world, often through new venture creation. So we partner with the Engine, we partner with Martin Trust Center, we partner with Deshpande Center. We're particularly interested in catalyzing discovery. I'll say more about this through seed grants. So we're often providing seed grants to different folks. I'll say more about nanoscience, but here are some of the topics that have bubbled up across campus. The seeds in our case really require interdisciplinary teams, people from different departments, different DLCIs. And moving forward into year 2, we're working with industry partners to issue grant challenges to the community. And then from an impact perspective, we're hoping that we can grow some of these seeds that we're planting literally today on October 1st. Our seed grants start today into strategic initiatives. We have some that already exist. I'll say a little bit about the MIT MGB seed program and the MIT program for health sciences and semiconductors. So HEALS across MIT, I was really shocked when I learned that 75% of the MIT community has already published a paper in health and life sciences. That's really remarkable. We're a very large community spanning, like I said, all the schools. As of this weekend, even I should say, HEALS launched on December 4th, but we've really been in existence for 6 months. And as of this weekend, we reached the milestone of having funded 140 different labs on campus through a variety of mechanisms, including fellowships, seed grants, et cetera. And if you're interested in the health and life sciences, please mark your calendar. We'll be holding an annual symposium in March. So I figured I would say a little bit about these seed grants that are starting today. I don't have cool images yet because the research is literally starting today. But of the 19 seed grants that we issued, 13 of them self-identified as being in the nanotechnology space. So just listing some of these, we have just one on the top is a collaborative project thinking about mapping multi-body, multi-organ dynamics of the maternal-fetal system. Nanotechnology plays a role in this. And we have colleagues from ECS, materials science, BCS, mechanical engineering, and civil and environmental engineering as part of that team. There are a number of projects that are associated with drug delivery and sensing and imaging. So we actually think within the HEALS umbrella, we can create a cohort of teams that are thinking about nanotechnology and work with nano. I'll say something briefly about the MIT MGB seed program that's run by Alex Jalick and Emery Brown. The goal of this program is really to plant seeds with our colleagues at the local area hospitals, focusing on MGB. They issued a call thinking more about AI-enabled diagnostics and digital health solutions. They got 134 proposals. They've now narrowed it down to 32 finalists. I got a peek at who those finalists are, and I would say probably 75% of those proposals relate to nanoscience. So that's really cool. And then finally, the MIT Health Sciences and Semiconductors program is another program that I think people here should know about. They ran their first grant cycle over the summer. Their strategic areas of focus are biosensing imaging, micro and nanofluidics, magnetic diagnostics, and ultrasound technologies. These projects require partnership with IMEC to actually move forward. And they were able to fund 4 grants in this area over the summer, and they'll have future runs as well. And then in the spirit of entrepreneurship, I'll just say there's an upcoming grant call, which is called the Momentum Grant. This is co-sponsored between MIT HEALS, Climate, and the Deshpande Center. And here what we're doing is giving a little bit of money for people to sort of check the boxes on internal academic work, with the idea that this would let you know if there's a there for commercial translation downstream. So these applications are due October 15th. We have a HEALS leadership team. If you have questions, thoughts, we would love to hear from you. There's an email address. If you send us a question, I promise you I'll get the email, and I'm happy to answer. And with that, I'll transition to the real show, which is the amazing science that our speakers are going to talk about. As I said, we have Anna Jaklenec from the Koch Institute, and she's going to talk about next generation mRNA delivery powered by predictive AI. Then we'll transition to Gio Traverso, who will talk about engineering for extremes, nano for health. And then we'll close out with Jagesh Shah, who is at Mirai Bio. He will talk about targeted lipid nanoparticles for extrahepatic delivery. Once we're done with the talks, we'll basically sit down for panel and take questions from the audience. So with that, I'll bring in Anna.
Well, thank you so much, Angela, and really a privilege to be here today and talk with all of you. Hopefully, my slides will work because I'm not sure how to give a data talk without slides. Excellent. So I want to talk a little bit about kind of our healthcare and medicine, how it's really changed over the last 30 plus years. I think we've really transitioned from treating illness when it occurs, sort of when you're bleeding, you go to the hospital and you get treatment, to really thinking about how can we prevent illness from happening in the first place. And I think it sort of started in the 90s and has really, I think, expanded greatly in the last 10 years or so. And I think part of the things that have made this possible is not just advancements in biotechnology and science, but also personalized and digital health that allows, and really information that allows all of us to really understand disease progression and understand things that we can do to prevent it from happening. Nutrition and microbiome science also is a big part of that. So there are many tools that we use for this, and I think we're all familiar with things like vaccines. I want to highlight one thing, and work at the Cancer Institute. About 13% of cancers are actually caused by infectious disease, and some of which can be prevented with vaccines. HPV is a great example, and I'll talk about that a little more in a second. HPV is a virus that infectious disease that leads to cervical cancer in women. Of course, early cancer interception is very common. We all have screenings, whether it's through imaging or other, Gio may talk about colonoscopies later, but there's many different ways to screen for that. And of course, nutrition and metabolic health is becoming extremely important. And I think we all understand why this is important. It can reduce healthcare costs, but most importantly, we can have healthier, better lives and longer lives as well. And so just to go back quickly to the HPV example, we know that HPV causes cervical cancer, and we know that vaccines now we have data from the CDC that show that the vaccines that were developed against HPV can help prevent that cancer from occurring, which I think is really a huge finding. But the issue still remains, as you can see in the graph here, that we're not immunizing. Not everyone is getting those immunizations, and there's still quite a bit of people that might be not protected against this disease. It's actually, if you're HIV positive, it can be worse. So what I've spent my effort really is trying to understand what are some of the challenges that persist there. And globally, there's estimates that go up to as high as 40% of people who start multi-dose vaccinations don't complete them, which means they're not protected. And even when we look at cancer therapeutic vaccines, which we all know might be the next great immunotherapy to treat cancer, oncology patients also drop out, and as low as 50% do not adhere to their full dosing regimen. So there's really a huge need to make these things easier for people to get the therapies that could really be life-changing. And so I've devoted a big part of my career in trying to address these issues. Of course, there's societal issues, but really from the technology standpoint, is there a way for us to lower the bar for people to get these therapies? And I certainly do a lot of work in the global health space and collaborate with the Gates Foundation. And in those cases, we're talking about low to middle income countries that don't have the infrastructure we have here. But also in the United States, there are areas, once you go outside of Boston and big city centers, that are rural and that just don't have the same infrastructure. I think sometimes we take for granted that we live in an area with some of the best hospitals and medical care in the world. And so those people may not have access to many of the therapies as well. So our group really tries to develop new technologies and new manufacturing to improve access and delivery of both preventative and therapeutic medicines for people across the globe, including here. We also do a lot of work in the area of microplastics, and micronutrients and nutrition. But today I'm going to focus on the two platforms that we developed for one, to extend the efficacy and durability of vaccines or therapeutics, and two, to make the ease of administration easier. So first, quickly, I just want to talk briefly about how we developed what we called our SEAL platform. And really the idea here was, can we develop a single injection formulation that for vaccines that are self-boosting, so you don't need to have the repeated shots. So this can have improved efficacy and it can be durable from that one single injection. And the idea is, as you know, there's many different vaccines. Now we have the mRNA LNP vaccines, which I'll get to in a minute, but there's different types of vaccines, different type of molecules that need to deliver. And so we decided that we wanted a platform solution, one that could be applied across the different modalities of vaccines or molecules. And actually, as Professor Bulovich echoed, to sort of echo what Professor Bulovich said earlier, we ended up really looking at the semiconductor industry and how they can package things in tiny little things, because we wanted to essentially build a tiny little cup that could take any drug and could be injected and delivered, and then it releases that drug at some point in time after it's injected. The Nano lab was just instrumental in helping us develop this technology. There's actually a dirty room where they let us play around with various things to make this work. We ended up publishing this work in Science, and really what we did is, until that point, this was in 2017, the drug delivery field, really with the drugs that were to be delivered were usually, for long-term delivery, were placed in these matrices, and the drug was throughout the matrix. That limited the type of control you can have with regard to drug kinetics. In the case of vaccines, you need the vaccine to be stable and then released months after you inject it. And so to really have that kind of precision and control over the kinetics, we went to a core shell structure, or we wanted to design a core shell structure. Just like an egg has a shell on it and the egg is stable, but once the shell breaks, it comes out very quickly, as we all know. So that was really what we wanted to create, but at a scale so tiny that could be injected with a regular needle and syringe. And so it was very difficult to do this. We failed, for those students in the audience, we failed many, many, many times. But eventually we created this new manufacturing process. It was almost a new way of 3D printing where we can make these bases that are made out of material, medically approved material. And then we use piezoelectric nozzles, which are found in inkjet printers, to fill it with any drug or vaccine you can imagine. And then we seal these particles. So we created this core shell structure where the drug is in the center, and it's stable until that shell breaks, just like an egg. And we were able to achieve this using the MIT.nano facilities. And today we have a company that's now scaling this process. And if you can see, this little 3-inch wafer has about 7,000 of those particles in it. So we really took something that you can do in microelectronics and semiconductor industry and brought it to the pharmaceutical industry. And this allows you for these very precise kinetics. And this is just what the particles look like. You have an SEM on the bottom, and you can make these with high fidelity in these large arrays. So it's really a beautifully manufactured process, just like you have in the semiconductor industry. But most importantly, we're able to, each vertical line here in this graph shows in vivo release of the drug inside or vaccine after it's injected. And you can see we can not only release days after injection, but months. That last one on the right is 6 months out. And the way we can do this is the material of the shell is from PLGA polymer, which is a polymer used in the medical industry as resorbable sutures, as screws. It's used in some drug formulations, so it has a huge record of safety. And we can tailor this release to whatever is needed, and we can mix and match these particles. There's a lot of data I could show, but I just want to highlight that we've shown that we could use these particles from a single injection to deliver 3 doses of the polio vaccine. We've shown that in non-human primates, you can get protective titers. And we've shown we could do this for many different types of vaccines: hepatitis B, which is a recombinant protein vaccine, influenza, and of course mRNA COVID vaccines, and I'll get to that later. But hepatitis B and poliovirus vaccines have completed non-human primate studies. We've also shown that you could use this technology to treat other diseases. You could use this for cancer immunotherapy drugs. We have, again, all published data. We published in Science Translational Medicine. You can use it as photo and chemotherapy drug to treat metastatic cancers. And also, you can also use it to leave drugs behind after open heart surgery on the heart to help the heart tissue regenerate. This was just accepted to Cell Biomaterials. And so we've shown that we can do all these really beautiful things to extend the durability from that single injection, single time of contact with the patient. The other thing that we're really interested in is really to make the ease of administration of drugs and vaccines easier. And for that, we went to microneedle technology. And I don't know if those of you that are familiar with microneedles, they're kind of a bed of nails, a little Band-Aid that you can press into the skin and releases the drug in the tips. It's actually made from sugars. And as we know, during the COVID pandemic, there was really an issue with delivering the vaccines, the storage, and so on. These little microneedles are in solid format, and they don't need to be refrigerated, so they're easy to handle. And here's one, for those of you who probably can't even see it, and that could administer your dose of vaccine or any other a drug if that's the case, and you can just press it onto your skin. And so what we did is we designed and built these three-dimensional printers that could be shipped basically around the world to it doesn't even have to be a pharmacy, so it could be shipped around the world, and the ink contains the vaccine as well as the material to make the microneedles, and so they could be made on demand. For that, we had to come up with a new manufacturing process to fit it into a box, which you see there in the center. If you look, there's a centrifuge-free mold filling, so we were able to build that into these printers, and you can, again, build these arrays of these microneedles that can be delivered. I'm not going into all the detail, this was published in Nature Biotechnology for the sake of time, but we've shown that these are a lot more stable at room temperature. They contained mRNA LNP vaccines. We did this work during COVID, and we showed that much more stable at room temperature than the conventional vaccines that are in liquid format. In fact, we had 6-month stability at room temperature with these solid state formulations. So to just sort of, but the challenge remained for us to create these, we had to develop a new LNP, a new nanoparticle that was stable under these conditions. It has to survive the manufacturing process, which involves drying, and then it has to be stable in storage conditions, and in the first technologies, it has to be stable in the body for a long time as well. So my students got a little frustrated, and postdocs, in trying to formulate some of these things. And like I said, we had this really great formulation, but what we wanted after all this work was done, people called us and they want us to work with some of the clinically approved vaccines and formulations from companies like Moderna and Pfizer. And what we found is that the Moderna and Pfizer LNPs, when they go through our processes for these two technologies, they don't survive. And so we had to really think of this from a formulation perspective. And so we developed, in collaboration with Professor Mina Konakovic at the Computer Science Department, we created a machine learning accelerated program that allow us to really test all these different formulations. And this is an example. What it did, it would tell us what excipients to test. We had in a small 96-well plate, we would dry the samples and see which ones survived, and we'd feed that information back into the algorithm. And then once we had the lead formulations, we would then test them in our devices and eventually in animals. And so very quickly, again, we worked with Moderna and Pfizer vaccines, and this really shows you the progress of our optimization algorithm and the various rounds that we tested. At the top there shows you the various rounds. Our target was to reach 100% stability compared to the liquid formulation. The bottom shows you the various compositions that we tested. And you can see after 6 rounds, it's not a linear process, but we reached 100%. Again, we characterize these various aspects, and I'm happy to, for sake of time, I can't go into it, but we can talk afterwards. And then we tested it in 2 models. One, we went to the mouse model, and we validated that, in fact, the AI-assisted formulation ended up performing just as good as the regular injectable vaccine. So it really validated that this works in a mouse, and of course, a lot of things work in the mouse. So, and again, just more data reiterating that. So then we went to non-human primate model to see if it works there, which is closer to human, as we all know. And again, we had these brilliant results that showed that you get the exact same immune responses if you use the solid state formulations with the AI optimized formulation versus the liquid stuff that you can get from Moderna or Pfizer. So this was really thrilling. But what I wanted to end on here and highlight that the development timeline of AI assisted mAb formulations was really tremendously shorter than anything else we've done. So first we started doing this process manually. It took over 4 months. We explored 100 different excipients, and no formulations matched. So we failed. After 4 months, we failed and could not have a formulation that worked as well as it needed to. And so then we decided to try a different approach and started using the AI, implemented the AI model, and within less than a month, we had our lead formulation, which I just showed data for. And then after that, we went into rodents and finally NHPs. So with that, I just want to emphasize how powerful some of these tools are that we have in accelerating our progress. And then I also want to highlight how great MIT.nano has been to us. It's really allowed us to innovate in so many different spaces and really bring this semiconductor microelectronics aspect into the pharmaceutical space and really to advance healthcare for global health. And then most importantly, I want to thank our really terrific staff of scientists, in particular the work I showed today, Jimby and Khan. Jimby's a graduate student in Course 6, and Khan is a postdoc in our lab who really have been tremendous in doing a lot of this work, and of course, all of our funding agencies as well, including the Gates Foundation and BARDA. So with that, I thank you.
Let me see if our slides are, I see them on the screen. All right, well, wonderful to meet you. I'm Gio Traverso. As you heard earlier, I'm in the Department of Mechanical Engineering. I'm also a gastroenterologist, and I practice across the river over at the Brigham. And what I'd like to do over the next 10 minutes or so is share with you 3 recent stories, really speaking to the nano sort of aspect and how it relates to health, but really across length scales. And so with that, let's dive right in. And we tend to focus on the GI tract, and hence the picture of the pill. So the first body of work is work that was led by 2 incredible postdoctoral scientists in our group, Alvin Chan and Ameya Kirtane, who are now faculty members in Singapore at NTU, as well as in Minnesota, respectively. And the challenge that they took on was really thinking about how to accelerate the development of nanoparticles for delivery of nucleic acids, including mRNA and others. And this is work that has been supported by several bodies, including ARPA-H, as well as the Marble Center here, as well as the Singaporean government. And how we refer to this system is as COMET, in that it is a transformer-based model that really helps accelerate the whole development process. But let me just take a step back for a second and remind you of the challenge that we have in front of us. So mRNA is a nucleic acid. If one injects this nucleic acid into the body, it is unable to be taken up by cells usually and or degrades fairly rapidly. And so somehow we need some help from a lipid nanoparticle. And that carrier or vehicle is what enables essentially entry into the cell. Traditional LNPs are composed of a set of ingredients. And so here we have some of the more common ingredients. There's an ionizable lipid, a helper lipid, a PEG-based lipid, and cholesterol. And very fine sort of changes in the ratios of each have incredible effects on the ability of that nanoparticle to enter the cell or even to target different parts of the body. And so recognizing that there's this tremendous sort of impact on both delivery, but also the site of delivery. What Alvin and Ameya and the team decided to do was initially build a very large set of experimental formulations, test them, and then use that data to then train a transformer-based model that really could treat each individual component on its own, but then use that framework to then help accelerate the next step. And so they were successful in doing this. This paper just came out in Nature.nano just a few months ago. And this is just to give you a flavor of the kind of things that one is able to achieve very quickly through these in silico generated kind of formulations. So I include here data from the rodent work, and we have in green the clinical benchmarks, and then in the light blue, the level of expression of the amount of protein that is produced in vivo, and what we're seeing there are improvements that are greater than 5 to 40-fold above the clinical benchmarks. But now we have the ability to very quickly arrive essentially at the answer by using that framework. Now, I'm going to shift gears a little bit. I'm going to go now to the macro, but you'll see how macro enables and actually works together with a nano. And this is work that we've been doing now for about a decade, and we've had collaboration with Novo Nordisk, the NIH, but also ARPA-H. There have been many people involved, and these are some of our students and collaborators over this journey. And the fundamental challenge here that we were tackling is the recognition that most people, if they have to take a drug, would rather take it through the route or the system that is shown here on the right, a capsule, over the injectable format here on the left. Now, the challenge, of course, is that many drugs, like insulin or Humira or even mRNA vaccines, is that they are degraded in the GI tract. And that's because our GI tract has evolved to degrade these materials. Many of our foods are made of proteins and nucleic acids, and we've evolved to degrade these. But also, these molecules are so big that they're unable to essentially transport or travel across the GI mucosa. And so what we did, really inspired by, in this case, by cephalopods, squid, was think about systems that could use jets to really facilitate that injection. We had done a lot of early work using different tiny little needles, but we said, maybe we can eliminate needles from this process. And someone can click on the screen. Perfect, thanks. And so what we did was we developed a family of devices that facilitated delivery of these jets. And these jets essentially carry nanoparticles through the tissue, and then these are able to distribute to generate the therapy that is intended for the specific condition. And so having developed a family of devices, let me just show you what this looks like. So this is one of those sets of devices. Essentially, they use a jet, and what you'll see here is essentially that jet sort of delivering that stream of nanoparticles. In this case, this is on the bench, but this also happens in vivo. And to show you what happens in vivo, here we have cross-sectional imaging of delivery in different parts of the GI tract, in this case of a pig. But what we were able to do was quantify the fundamental pressures required to really inform the optimal delivery parameters in different parts of the GI tract. So having established all of this, we looked at a few different therapies that typically are not administrable through the oral route. We looked at, for example, GLP-1 receptor analogs, but now we're achieving bioavailabilities or levels that are comparable to an injection. So there is an oral semaglutide on the market. That bioavailability is 1% to 2% in that formulation. Here we're achieving 50 to 90% bioavailabilities that are comparable to subcutaneous administration. We also demonstrated the ability to deliver siRNA using these systems. So I'm going to transition. I should say that that body of work was just published at the end of last year in Nature. And then this body of work that's been supported by ARPA-H and the Gates Foundation and led by an extremely talented scientist in our group, Troy Kang, really set out to tackle a fundamental challenge of initially adhesion or engagement with wet surfaces. So certainly I think we recognize that on the wearable side, but this is a big problem in the GI tract where we have a wet, moist system or surface that is turning over on a regular basis. And so we said, well, maybe we can look at nature for inspiration. And essentially what Troy did together with our collaborators was look at a fish or a family of fish called remora or suckerfish. And these are fish that essentially are able to adhere to many substrates. And so on the right of the slide, you can see the range of substrates, right, in the form of whether it's a shark or a turtle. And these fish have evolved to adhere to these different surfaces. And so we said, you know, maybe there are some lessons that we can take from this evolutionary experiment. And if I hope you can appreciate there the sucker that has been highlighted, essentially you can see that there are a lot of similarities, even though the substrates are actually quite different. And so what Troy, together with our collaborators, did, we were able to get remora and study essentially the sucker system to really understand how this system can integrate and facilitate that engagement with the mucosa. We also filmed them, so we collaborated with a group at BC, and we were able to film the engagement with synthetic sort of gastric tissue to really understand the steps involved in how this suckerfish can engage with the surface, and studied and really delineated all the individual steps of that process and developed a synthetic remora, a capsule that can do all of this. And so we demonstrated that not only could we do this, but we could recapitulate that adhesive system. And this is, of course, at the macro scale. And what the system allows is essentially you can administer this, and the natural peristaltic motion in the GI tract essentially is acting really to help with the engagement with the mucosa. So you'll see here, there is a contraction in the esophagus, and then the system is adherent to the GI tract. Now, this is where nano comes into the conversation, where now we have a macro structure enabling that juxtaposition of the device with the tissue or the site of delivery, and now we're able to load these devices with nano formulations. In this initial example of an antiretroviral for the treatment of HIV, where we're able to now support treatment orally with a once-a-week capsule. And beyond that, we've also delivered mRNA using these systems. So similar to some of the things you just heard from Anna, we've developed some solid formulations that we're able to immobilize on these systems. And not only are we able to immobilize them, but we're able to achieve delivery and functional expression in pigs using these systems. So hopefully it gives you a little bit of the flavor of what we're doing in nano and bridging some of the medical device macro sort of aspects and how we are at least reimagining what we can do for patients. I want to thank the work of many, and these are some of the folks and their families at our recent celebration on one of the Boston Islands. And with that, in case I don't get to your questions, here's my email. I want to acknowledge all of our incredible institutions, but of course, all of the funders and sponsors that really enable and support our work. So thank you so much.
while my slides are coming up, I put in the fact that I'm of you, PhD 99, so, but spent some time at institution up the way, then transitioned into industry. So one of the through lines that I'll take advantage of what Anna and Gio talked about was the idea that we can deliver mRNA, which is the language of the cell, to reprogram cells. We can get them to make vaccines so that they can present antigens so that we can generate an immune response, but we can also get them to reprogram in a variety of different ways, including, if you're familiar with CAR T cells, so that the CAR T cells can go and kill cancer. So I'm going to give you a flavor for how we're trying to get to these other cell types using lipid nanoparticle technology that was leveraged during the pandemic. So Mirai is actually a flagship company, and I'll give you a bit of a flavor of our business model first. We do not make our own therapeutics. We are a foundry of sorts. We generate targeted lipid nanoparticles that go to a variety of sites that I'll describe and try to identify partners that have therapeutic cargoes or therapeutic hypotheses that would then leverage our ability to be able to get to those sites. So our work is really very broad in the context of trying to get our nanoparticles to a variety of different tissues and then enable therapeutics for the next stage. Our model is really built on, another through line here is AI and the use of those tools, but lipid nanoparticles, as Gio described, is a chemistry game right now. It's really understanding what kind of ionizable lipids, which are thought to be the linchpin of the nanoparticle, but also other components that can be put together and then screened at scale. So how do we screen them at scale? So part of what we've developed at Mirai is a microfluidic technology that allows us to make large numbers of different LNP formulations all at once, and then be able to put many of those into an animal, into a single animal, and then deconvolve and multiplex where each one of these formulations has gone. And it'll be a technology that many of you will be familiar with, a barcoding technology that allows us to not understand just where the nanoparticle went, but as you saw in Gio's animation, where it actually delivered to the cell. So where it actually got out of the endosome, which I think of as the lipid nanoparticle jail of the cell, how can it escape so that it can have its therapeutic effect. One of the other, when we carry out those in vivo multiplexing, we generate a lot of data. That data allows us then to refine where our nanoparticles can go, and this is where the active learning-based approaches based on this in vivo data has really dramatically increased our potential for getting to cells of interest. We're a part of the flagship ecosystem, and leveraging that allows us to also look at things like binders. So once you can get a nanoparticle to go there by chemistry, don't forget about biology. Biology has binders and antibodies that can actually even increase the level of potency. So we're bringing those two technologies together to be able to get high potency delivery to cell types of interest. And I think we've heard a lot about scale here, and being able to do something at the bench top, being able to do something in mice, really does ultimately is just the aperture that you need to open up to be able to get to preclinical studies in monkeys, but also into human beings. So how do we make sure that we can scale that material in the context of clinical and commercial production, GMP quality? And that's where we have some partnerships with our industry partners like Thermo. There we've been able to now take our partners all the way from the discovery element all the way to clinical evaluation, and hopefully their success that would result in commercialization. So I'm going to talk about the elements of the platform that we've built and then give you an example of how we've built a particular targeted nanoparticle for our goal. So we have some chemistry. I think that this is kind of the table stakes in LNP biology, is trying to understand kind of what kinds of ionizable and other components you can put together. We've developed this high throughput formulation platform internally, a proprietary system to be able to get 96 nanoparticles of different formulations simultaneously. We've also developed a proprietary barcoding system. As I'll describe, it allows you to distinguish from being just getting to the organ of interest, but actually getting into the cell type of interest and escaping the endosome and delivering that therapeutic capability. And then machine learning for us is a tool, and we try to sprinkle it in wherever we can, and to give us the advantage of being able to take large-scale data sets and turn them into new learnings. Another piece of the flagship ecosystem that I just wanted to highlight here is mRNA is just one language that the cell speaks in. There are a variety of different languages of nucleic acids that the cell uses, and we've been able to work with a large number of different kinds of nucleic acids, mRNA, transfer RNAs, small interfering RNAs that you've heard about, as well as DNA and gene editing and self-amplifying RNAs that are quite large and still remain to be are challenging to formulate. But we've been able to expose our lipid nanoparticles to those as well, to be able to understand what other languages can we deliver and what other therapeutic hypotheses can we deliver to the cell. So our pipeline looks quite broad, but really what we're trying to do is unlock tissues so that we can enable these therapeutic hypotheses to be developed. So I'll talk a little bit about how we have built out our first set of components. We usually start by identifying a cell type of interest. The base LNP is really where most of the work needs to be done, being able to ensure that the particle gets to the cells and tissues of interest and then doesn't get into the cells where you want to avoid off-target toxicity. Then additional, as I'll show you, there's some benefit to adding some intelligence to the particle, as it were, by adding in binders that then selectively get to cell types of interest and revealing that. So I'll just describe our barcoding technology first. So we have a large library of proprietary lipids. And what we do is we formulate, in this case, example is 10 lipids. So this is real data. So it's not, even though it looks all very colorful, it's actual data. We took 10 ionizable lipids. They are now encoded by 10 different nucleic acid barcodes. And what you see on the far right, your left, is the dose. So we're trying to dose in equal numbers of these barcodes and different different lipid nanoparticles. What you see in the middle is now we're very interested in getting to immune cells for this particular example. So we're pulling out the spleen and then asking how many of those dosed barcodes actually ended up in the spleen. So that allows you to look at now the proportion in the stacked column plot of all the different barcodes that ended up there. This is a technology that has been used pretty widely, but one thing that kind of causes some consternation, at least in our experience, is that only about 1 to 2% of those LNPs actually deliver their therapeutic cargo into the cytoplasm. So many of these barcodes are stuck inside the cell. What we do in the last part is that the nucleic acid that then gets delivered into the cytoplasm undergoes a modification that changes its sequence. So we now can distinguish between the cytoplasmically delivered material and the material that was dosed into the animal. And that allows us to look at different kinds of phenotypes. So enrichment here, we're seeing that an LNP gets well into the spleen and also seems to get well into the splenic cell cytoplasm. That's what the third column would be. There's also some examples where the reduction in the biodistribution turns out to be very small in the spleen itself, but it's very large in the cytoplasm. These are particles that now we've done a good deal of analysis on, actually have increased endosomal escape. If you put them in the endosome, they deliver their material very well, so much so that they don't even hang around in the endosome for a long time, and as a result have very good delivery into the cell types of interest. And then, of course, there are nanoparticles that we can exclude. They don't really go to the spleen very well, and they don't really accumulate in the spleen. So this allows us then to identify a set of lipid nanoparticles that can go to, in this case, the spleen, which is the second column here. One critical piece of the lipid nanoparticle field is how do we detarget the liver? So those of you that took your physiology, you can't detarget the liver. It takes 25% of the volume of the blood. So you can't get particles not to go there, but you can get them not to go into hepatocytes. And that's part of what we've developed here, is a number of lipid nanoparticles that don't undergo delivery into the liver, so that then they can persist and go to other sites in the body. And what we've seen here is 2 different nanoparticles here that have modest levels of reduction in liver delivery and some increased levels of the spleen. And this is our base for our ability to then do active learning-based formulation optimization. So what we do is we take our formulation, and luckily Gio described what the 4 components are that go into traditional nanoparticles. They're put together in a particular level of formulation components. We then put those into animals at large numbers, and then identify all of that data to try to understand how we can hill climb and increase the level of on-target delivery in the spleen and reduce the level of delivery in the liver. And what you can see here is, over the course of a variety large number of in vivo studies, we can, on the right-hand side, sorry, your left-hand side, you can see that we're minimizing liver delivery. In this case, almost 2 orders of magnitude in the context of a bioluminescent cargo. And on the right-hand side, we've been able to maintain delivery to the extrahepatic site. And so we started with our barcoding, allowed us to identify a base LNP that was the substrate for machine learning, then allowed us to then look at delivery. And so what you can see on the bar graph here is delivery now to human cells in a mouse where we've put in human T cells. And what you're looking at is you see that our commercial lipid gets to about 30% of those T cells, and we can increase the level of delivery to about 60% in this model. But perhaps more importantly, the immunohistochemistry, which is a more qualitative readout, shows you that the commercial lipid gets avidly to the liver. And this is measured by the brown staining, which is a readout for the reporter that we've delivered to these cells. And you can see that our Mirai lipids have remarkably lower levels of delivery. So we've now created a particle that, when injected IV, will go to T cells and not to the liver. That wasn't good enough. We wanted to see if we could do even better, so we identified a number of receptors on T cells. You're familiar, those of you that are familiar with the alphabet soup of T cells will know CD3, CD8, CD4, but more importantly, we have a number of companies, again, within the flagship ecosystem that we love to take advantage of, that are building many kinds of binders, traditional vaccination or immunization-based approaches, but also ones that are based on viral proteins that have been evolved over millions of years and that have low immunogenicity and high affinity, on the order of picomolar binding, some of these viral proteins. And then also, you may be familiar with Lila Sciences, recently had a fairly significant A round. They are building machine learning binders for us, and they happen in a month. And you don't have to wait for any mice to kind of develop antibodies for any of this work. So we're very excited about the opportunities to build these binders. One of these binders against CD8 was then added to the surface of our lipid nanoparticle, and I'll just quickly walk through the fact that what we've been able to do is take the base LNP and now increase the level of delivery dramatically inside these mouse models and maintain the level of off-target delivery compared to our commercial LNP. And this is our base substrate for now doing some clinical studies with some partners. Those of you that are familiar with the CAR T field will know that in vivo reprogramming of T cells to be able to not have to take the T cells out for each patient and then put them back in again, but actually directly reprogram it is actually a particular case study here, and we hope to have some NHP data on that very soon. We've taken the same approach, and now we've been able to go after other immune cells and hematopoietic cells, stem cells as well, to be able to direct a lipid nanoparticle with a base to a cell type of interest using one of these base LNPs as well as a binder. So we've developed a number of technologies. Happy to discuss those with others. They may be, that may be of interest to you. Multiplex barcoding in vivo, the machine learning, the active learning, the immune cell tropism, as well as our modular extrahepatic platform using a binder. Again, our model is really co-creation with a partner. So we currently have a number of partnerships where we're looking at a variety of different therapeutic cargoes using these targeted LNPs, and we've opened up a number of these cell types. Some of them may be of interest to people in the audience. Happy to discuss. So thank you very much.
Can you hear me? That was great. And we're going to have questions from the audience, but before we do that, today has been an interesting morning. We started the day off with a government shutdown, and this is sort of illustrative of the sort of impacts that we're seeing across R&D. I often like to tell people that the model in this country for R&D is going to change and evolve in many ways. That affects all the sectors. And given that we have 2 academics and 1 former academic who's made the jump into industry, I'm curious to hear your perspectives about how this will impact you in your respective sectors.
You want me to start? Well, of course, it's not good from many different levels. I think it's really hard for us. While we are very good at controlling experiments, it's really hard for us to control the government and what's happening around us. So I think there's really not much that we can do outside of just trying to manage our lab and our spending and sort of how we think about making sure that we continue to innovate and continue to push the work forward. I think actually at the beginning in your talk really illustrated that MIT, given all the uncertainty around funding, not just due to the shutdown, but MIT has really put forward a lot of different programs to kind of help supplement maybe some of the losses that we'll see, and that we are seeing from the government funding. The other aspects are looking at other ways to receive funding, and that's foundations. And myself, and I know Gio, we've been fortunate to work with the Gates Foundation and other foundations that I think are not affected necessarily, though obviously they're being asked to fund more work, so that's affecting how much they can give, right, to some extent. And then, really being creative, and maybe Jack can talk more about that, how we can partner with industry, perhaps some sort of in-kind things where there's an exchange of scientists or access to different facilities. So really trying to make things happen even though you don't have that grant coming in, but maybe you can have a free person, if you will, and free access to equipment or something that can really help move the research forward even though you don't have the big grants coming in.
I mean, Gio, you've talked a lot about ARPA-H. So do you expect any sort of impacts on your group?
So with respect to ARPA-H, I received our ARPA-H grants. I don't know if people are familiar with ARPA-H, but ARPA-H sort of is a new entity, relatively new, that's sort of about 3 years ago, to basically support high-risk, potentially high-reward concepts and to try and nurture them, all the way through, ideally through early-stage clinical trials. And our ARPA-H is one of the largest ones in the country, over $60 million, aiming to transform what we can deliver orally. And so far as can we deliver nucleic acids and achieve high bioavailability, or can we deliver electroceuticals, which wasn't something I talked about today, but a lot of the work that we're doing now. And so I received a notice yesterday that our funding happens to be managed by the Department of the Interior. That's just a nuance of how the different funding comes through. But I received a notice that we should continue. So our guidance was continue, and that was the guidance. I think just to add to what Anna was just saying, and I think whether there is a shutdown or not, I think if anything, it just highlights what I think increasingly we as an academic community recognize is that we need to diversify if we haven't done so already as far as our funding streams, right? And so as Anna said, we've been fortunate to work with the Gates Foundation for over a decade, and I've worked very closely with Novo Nordisk for over a decade as well, and a few others. And I think, if anything, whenever there are shutdowns, it's not the first one, at least unfortunately, in the last few years. And I think it just again sort of reemphasizes the need as an academic to try and at least seek out opportunities and partnerships. And I mean, that's my two
And then
cents.
how's this impacting industry? And is this like maybe is the silver lining that we may all play better together?
I think we play well, pretty well already. I think I don't want to. It's been said many times, but not only do the ideas, the nascent ideas that are leveraged in the context of industry, whether by individual entrepreneurship or by licensing some of these ideas, you guys are generating the workforce. I love hiring MIT students. They're fantastic. They are well trained. So I think that we just it feels like there's no more toothpaste in the tube at some point, right? If we do not fill this pipeline, we're not going to have those kinds of people. But to your point, in the near term, let's say that we can get over this at some point. I do think that there are some real opportunities for really interfacing some of these things. I'll be honest, there's no chance I'm going to be getting any remora fish at Mirai, setting up some kind of lab there to look at those. So there are some really fantastic ideas that when academics are ready, we really would love to partner. And what do we offer? We offer some of the opportunities to take things to scale, perhaps, that maybe are not always available, the ability to be able to scale out in some large sets of experiments. So we do have capital, somewhat, that I think could be deployed. So everybody will have to give a little bit on either side. It's not the same kind of trench warfare that we might have experienced before, but I'm very excited about what we can do.
And you mentioned an interesting model system. So we also know that the federal government is changing how we think about the use of animals within research and trying to get us to think about sort of alternatives to traditional animal models. So how are you all thinking about that push?
Well, I think it'd be great if we didn't have to do all the animal work, and there was a very validated way to evaluate things in a non-animal model. I think, and so if that, some people are working on this and lab on a chip and various things. We're doing some stuff with women's health in that space, trying to see how drugs on lactating mothers get into the breast milk, and can we screen which drugs do that without in a dish rather than going to actual experiments in animals or people. And I think as long as it's safe, it's actually a good push. But there needs to be resources and funding to really develop and validate. That's really the key piece here, is validate that we have a good alternative that is predictive. And I think once we have that, it could be actually helpful. But of course, we don't want to, I certainly wouldn't want my child or myself to receive something that hasn't been tested in an animal just on a chip. But anyway, in a few decades maybe we'll have such strong validation that we'll have confidence that that is safe.
Gio, what can we do at MIT along these lines?
Yeah, I mean, I think that it's wonderful to see that sort of these things are aligning, and we've been working on ex vivo and in vitro models of the GI tract, for example, and the liver, for about a decade. I mean, we started a company that is based here and in Switzerland that, is doing this for drug delivery science. And also we have some more recent data really predicting toxicity in a dish that translates really well to large animal models. So I agree. I think that it's wise. I agree with Anna, though. I think that it's still early, right? So the vision and dream of having these in silico twins that are able to predict the outcome of the experiment without having done the experiment in vivo is challenging with the current data set. Can we get there? Perhaps, and I hope that we do. I think that would be incredible. I don't think people want to use animals, and I think that we really try to do everything we can before we get to that point, but I think it's more the burden of data to overcome that hurdle to convince the regulators and ourselves as the end users or loved ones, et cetera, to use it.
And is industry looking at MIT to sort of help fill the critical path in some way?
Absolutely. I think that I'm not going to add to what and Gio said. I think it's fantastic. I think the regulators are also thinking about the modularity of these drugs. So the idea that maybe for a single drug you'll have to do certain kinds of animal studies, but then if you just replace the mRNA would you have to do all of that work
Yeah.
again? And so this idea that we can then version, for the computer scientists in the audience, version control these drugs and then take advantage to reduce the burden of animal models in the aggregate, I think, is another area that where we can exploit. And again, MIT is not just the tech part or the entrepreneurship. It has strong binds into the regulatory agencies as well, because they all do work through kind of the logic and education that we all use. So how can we make that happen?
We have more questions on the list, but we have run out of time. So I'm sure our speakers would be happy to address any questions that you have at the break. But please join me in thanking them again for a great panel.